gosh. Thank you for that. Apparently I've set the... Sorry about that, everyone. I've set the privacy. Okay, so it should be fine now. Hi, everyone, and welcome to the first session of Varsity Sci, well, of our fourth day of the event. During this session, we will have two brilliant biochemistry talks. We will start off with Godwin, who will introduce us to artificial carbon dioxide fixation, of course, as you can see on your screens, using sustainable enzymatic synthesis. So that's a very hot topic. Artificial photosynthesis is an active area of research, and I'm sure that other methods of carbon dioxide fixation will be very useful in that area as well as in other areas of biochemistry. What we will have after that talk is we will have a talk by Susanne on the lipid-facing helix of pentameric ligand-gated ion channels. So that is going to be a very important subject of research for anyone who is interested in cell signaling, in receptors. And of course, during the current pandemic, receptors is something that we're all concerned with. Binding is something that we're all concerned with. So without any further ado, I'll hand over to Godwin, who will be our first speaker.
Thank you very much for your kind introduction. Yeah, just to introduce myself again, my name is Godwin Aleku. I am an early career research fellow at the Department of Biochemistry in Cambridge. Basically, what I do, I work on enzyme discovery and enzyme development, and developing an enzyme and enzyme system and seeing how we can apply this engineer enzymes or developed enzymes in organic synthesis. So my research spans the areas of synthetic organic chemistry to industrial biotechnology and synthetic biochemistry. Today, I will be discussing on one of of course, the sustainable ways of, or the clean way of making pharmaceutical or chemicals. And I'll be discussing on how we can utilize CO2 as a chemical feedstock to make chemical enzymatically. Now, one of the topical issues is the impact of our processes, our manufacturing processes on the environment. And of course, we recognize that there's a huge impact on anthropogenic human activities on environment. And this affects it has environmental and ecological impact. And there's a concerted effort to see how we can develop systems to make our processes green, and of course, to reduce impact on the environment. And some of the interventions have come from the automobile industry, actually. And of course, there's a huge encouragement to plant trees to be able to kind of reduce the impact of excess CO2 in our environment. One approach we could really do is, can we utilize the CO2 that is emitted from our processes? Can we recycle that and use it for either synthesis of chemicals, synthetic or green fuel and solvents? In this regard, there's been a lot of effort with regards to taking CO2 and reducing it to assess solvents like methanol, or you can get methane or dimethyl ether. But an interesting reaction is incorporating CO2 into an organic molecule, using CO2 as C1 building block, really. So you start with a simple starting material, and you can use CO2 to build a complex molecule. Now, it's interesting to know that CO2 fixation is one of the most prevalent reactions in nature. And so nature has developed pathways. So in autotropic pathways, like photosynthesis, you have CO2 fixation route. And this is one of the key reactions that is involved in making most of the organic frameworks in nature. And so nature has evolved a lot of CO2 fixing enzymes to catalyze the physiological reaction of CO2 fixation. However, when we want to take this enzyme to apply in synthetic substrate to fix CO2 in synthetic substrate, we find that these enzymes are generally inefficient. And a lot of people have tried to engineer this enzyme, like the ribiscose enzyme. We've seen a little success for the application of this enzyme. And besides, this enzyme normally would require expensive cofactors like ATP and sometimes unstable cofactors. So we're limited on how we can use natural CO2 fixing enzymes and application in synthetic chemistry. The other thing we could look at is decarboxylases in degradation pathways or in biosynthetic pathways. This other class of enzymes seems to be more robust and stable. However, the problem is most of this enzyme would prefer to remove CO2 from an organic molecule rather than fix CO2. So if you want to drive the reaction in the reverse direction, you have to devise strategies to overcome the thermodynamic limitations of this fixing CO2 into organic molecule. So with this background, we wanted to develop a system that first of all can have access to generic chemical platforms or scaffolds that are very prevalent in either medicinal products or just industrial products, really. And we needed to find a way of overcoming the thermodynamic restriction of fixing CO2. So normally how we will start this is, first of all, we will probably define what we call synthetic targets. And in this, we take into consideration chemical scaffolds or skeletons that are usually feature prevalently in either pharmaceuticals or specialty chemicals or bulk chemicals. Of course, if you're familiar with synthesis, you agree that alcohols, amides, amines, and carboxylic acids are some among the most prevalent functional groups in chemical industry, really. And so the next thing is to design a cascade pathway for which you can start from ideally simple starting material, easily sourced and simple starting material like aromatics like styrene, and of course CO2 as a feedstock at the moment. And then we could build a pathway to generate alcohols, so we generate amines, we generate amides, and we generate carboxylic acid. The next step would be to identify the right biocatalyst, and normally you have more than a step, and so you have to identify the right biocatalyst to catalyze each step of the reactions. And in this case, we envisage that the bottleneck will be actually identifying CO2 fixing enzymes that are efficient for our process. Of course, sometimes we need to develop this enzyme by direct evolutions, and then we can then assemble these enzymes, usually in one pot reaction, and seeing whether we can start from a simple starting material like this and assess a more complex molecules like that. So with regards to identifying the biocatalyst, we have a few options that we can consider. First of all, we'll probably want to explore what nature has evolved, what reactions are there, what enzymes are there in nature that catalyze, for example, the fixation of CO2. And we can look at the genomic context, genomic analysis. We can look at environmental DNA samples and functional metagenomic screening. But we can look at metabolic or biosynthetic pathways and probably look at enzymes in these pathways and isolate, maybe assemble enzyme from different pathways to form an artificial pathway. And this is what I'm going to be talking more about as a source of our enzyme. Once we've identified a template, we will sometimes need to improve the properties of this biocatalyst to fit industrial processes. We may need to increase the activity of this enzyme, the stability of this enzyme, the solvent tolerance of this enzyme. So we normally will use protein evolution, either a random approach, direct evolution, or we can use a rational approach of evolution. And there's been a lot of work where we can design a protein from a catalyst, from a protein catalyst from the scratch by protein design, but this is not trivial. So normally we would prefer to go the evolution route and the nature's diversity route. Once we have identified the right biocatalyst, we can then devise means of applying and optimizing the systems of applying this biocatalyst in organic synthesis. Right, so we decided to look at a biosynthetic pathway. Right, well, this work was done in Manchester, really, Manchester MIB. So when I joined the group of David Lace in MIB Manchester, there was two groundbreaking discoveries. First of all, the functional role of what we call the UBD decarboxylase, which is an important decarboxylase in ubiquinone biosynthesis. Some of you may know it as coenzyme Q. They identified remarkably a cofactor, a new cofactor called prenylated flavin. And this cofactor has an extra fourth member ring, six-member ring attached to the normal flavin skeleton. And this cofactor is usually active as the immune species. So the UBD enzymes, which are prevalent, of course, ubiquitous in ubiquinone biosynthesis or other respiratory quinones biosynthesis, catalyze the removal of CO2 from organic frameworks like this, the precursor of ubiquinone, to give you another intermediate before the ubiquinone step, ubiquinone itself. A closely related homologue, as they call the ferulic acid decarboxylases, and this you find them in yeast. They're related by function and sequence as well. This enzyme catalyzes the same reaction, but on a different type of substrates. So you would normally have acrylic acid derivatives like cinnamic acid being decarboxylated to give you styrene. Now, we chose to use this enzyme because they're normally stable and the cofactor is normally bound to the active side of the enzyme and it's catalytic. So we didn't need to add any expensive cofactor in our reactions. Normally, we've looked at the property of this enzyme. They can withstand some organic co-solvent as well. So we chose to use this enzyme as the starting point for our application. And so my next talk will be looking at these enzymes and the applications and with other partner enzymes as well to develop the pathway. Now, we had identified that enzyme as a potential biocatalyst for use, but we needed to overcome key issues. The first is that the enzyme substrate scope is limited to substrate acrylic acid derivatives. So for the enzyme to react to a substrate, you need to have the alkene linker between the R group, which can be an aromatic group, and the carboxylate. If you have substrate like benzoic acid that lack the alkene linker, the enzyme does not react with this kind of substrate, and so you don't find any product. So the question is, can we take this as a template to engineer this enzyme to assess more interesting scaffolds like the noctuic acid, indole, benzothiophene, and benzofuran? The second task will be to look at, of course, because of the thermodynamic issues, the enzyme prefers to remove CO2 rather than to go the other way. Practically, the reaction is meant to be reversible, but the equilibrium lies far to the right. So you will normally have the reaction go to the forward direction. How do we make this reaction? How do we reverse this reaction? How do we start from terminal alkenes and with CO2 and get the carboxylate? So I will be talking about the engineering aspect first, and then I'll talk about how we manage to circumvent the issue with around the equilibrium of the reaction. Right, so to engineer a protein, you probably have different approaches you can follow. Normally you consider things like, do we have enough good platform to screen wide range of enzymes? So you can either do, sometimes you probably want to look at a rational approach, you can have insights about the mechanism of the enzyme, the kinetics of the enzyme reaction or the enzymes, and understand sometimes the structural implications of the enzyme. And from that insight, you explore that to make changes on the enzyme, normally the active side, to achieve what you want. So we decided to go the semi-rational route where we combine insight from structural mechanistic understanding of the enzyme reaction and the couple with some elements of bias, some elements of random evolution. And I'll just talk you through that. So what we did was, this substrate that the enzyme do not accept, we term them as the inhibitors of the reaction. And this is the natural substrate of the reaction. So we set the carboxylation of this substrate as the model reaction. And we use microtiter plate screening to screen for inhibitors. So we monitor the rate of the reaction with different concentrations of this inhibitor. So we had about 3 aromatic acids. And once we identify competitive inhibitors, we examine the pattern of inhibition. And once we identify competitive inhibitors, of course, this is indicative that this substrate bind at the same active site with the natural substrate. We went further to confirm this using crystallographic studies. And so we obtained structures of the enzyme with the complexes of enzyme with a few inhibitors. And if you can follow me gradually, I'll try to walk you through this structure. So if you can look at the gray color is the natural substrate, and this is the catalytically relevant mode. If you look at the magenta one, which is the natric acid, which is we term as the inhibitor, and of course we identify this as the competitive inhibitor of the reaction. It aligns in a way that it's, we call it non-catalytic, it's a non-productive binding really. For the reaction to happen, this is the cofactor in yellow. You need this C1 prime of the cofactor to interact with the C alpha of the substrate. So this C alpha and the C1 must be brought in close proximity to enhance a covalent interaction. If you look at this substrate, the way it binds, the C alpha is too far away from the C1 prime of the cofactor. So in solution data, we don't see activity. Of course, we don't see, this explains the reason why we don't see activity. For this kind of substrate, the heteroaromatic, we found something very interesting. They assume 2 conformation. Most of the species binds in the non-active, non-catalytically active conformation. And if, some species bind in the catalytic active conformation. And in solution data, we could see very weak activity towards this heteroaromatic kind of compounds. So the one in this cyan color. And so from this insight, we could rationalize how we can have direction, how we can evolve this enzyme really. And we think it was an element of steric issues. And so if we try to address it from a steric hindrance point of view, we could potentially align this substrate in the catalytically active mode. And we hope that reaction will happen once the substrate is aligned with the right geometry. And so really you could think about making the active site residues around this area more bulky to push this substrate to align properly. Or you could make these ones around this area less bulky to allow the substrate to fall in and align properly. Now, we had also investigated through what we call alanine and serine scanning, the contributions of the active sites towards catalysis stability and sometimes specificity of this enzyme. And so we knew what residues to avoid, like the E2A2 glutamic acid, E2A2, and some other residues we avoided. And we chose from our preliminary understandings of the active site investigation, we chose leucine 185, methionine 283, and isoleucine 327. Now, to cut the long story short, we didn't find any improved hit with this library. So we made what we call saturation library, where the different amino acids are featured in the position, and then we screen the library. So that's the element of the random approach. We also looked at the saturation library of this, and this is what I'll be focusing on because we had some hits around the methionine 283 and isoleucine 327, sorry. We screened this using in vivo, so using whole cell. So we express this in E. coli, and we assess the activity for the decarboxylation of that. Now, E. coli whole cell does not decarboxylate this, so this allows us to monitor the reaction of the recombinant enzyme in whole cell. And remarkably, I told you that we observed some sort of activity with the heteroaromatic here with the wild type. We didn't see any activity with this benzothiophene kind of substrate. But remarkably, you could see that mutants or single point mutants at isoleucine 327S show remarkable activity towards the carboxylation of, for example, that. And of course, you can see the remarkable, the improved activity as well. And this is a novel activity because the wild type does not have activity. And we also identified mutants from the methionine residues, where if you change methionine to isoleucine, you have improved activity. And when you combine the two mutations, you have some slight improvement in the activity of a single mutant. For example, if you take that, which gives you 60% conversion to about 78% to 78% conversion with the double mutant. And we determined the specific activity for a number of the substrates. This we couldn't measure because there was no activity at all. And we could show that there were, just with single point mutation, there was about 50%, 150 times improvement in the efficiency of this enzyme to convert, for example, a benzofuran. So we wanted to see the rationale for the improved activity and whether it was, our hypothesis was right, whether it was a steric effect, whether that mutation caused the alignment of the substrate in the right direction. And we took the mutant that was very active and looked at the structure again with the substrate I talked about earlier. And you could see that, for example, if you look at the substrate that was in the wrong or catalytic inactive conformation, you could see that now it's aligned to the right conformation, and the C1 prime is in close proximity with the C alpha of the cofactor. The C1 prime of the cofactor is in close proximity with the C alpha of the substrate. And we, of course, because the bulky isoleucine 327 has been substituted with a smaller hydrophilic moiety, which is 7. And we could show that we could assess a wide range of heteroaromatics as well as unactivated aromatic compounds, achieving high conversions in some cases. And for the first time, we have shown that we have developed enzyme either. So at the moment, before this work, there was whatsoever no activity for no enzyme discovered to be able to decarboxylate naphthoic acid, this kind of skeleton benzenes. Though some groups have reported this in cultures, but there's not been any particular gene or enzyme identified to decarboxylate this. We've been able to show that we can start assessing this kind of moiety, although in low conversion. We also observed that the catalysis was radioselective. So if you have substrate with where the carboxylase used the third position, so 3 carboxylates, you absolutely don't have any activity. You don't have any activity for this kind of substrates. Of course, it prefers the selective towards 2 carboxylate kind of substrates, and that's important as well because it means that if we start from the heteroaromatic, we can selectively carboxylate at a particular site, site-specific carboxylation, and which is what we want in an enzyme system, really. So I've talked about the engineering aspect. I'm not just going to be talking about how we address the equilibrium issue with the reaction, and eventually we'll talk about the cascade where we apply this. So I did mention that the reaction lies to the decarboxylation step. So it prefers to go to it prefers to remove CO2. And so the key thing is, how do we push the reaction to go forward? Now, so we reason that if we can render this, or if we can convert this to another product, ideally a product that has more value than even this or that, we can potentially pull this reaction gradually to completion. Of course, if we reduce this to an aldehyde, that means this is withdrawn from the equilibrium, and it means that as species of this is formed, it's get withdrawn, and gradually you can pull everything from that to that. Of course, you could do alternative reaction. You can target the alkene bond and you can reduce that, and potentially you could get the saturated carboxylic acid, and you also remove this from the equilibrium as well. But we chose to look at the CAR system. So this is called carboxylic acid reductase. They reduce carboxylic acid to the corresponding aldehyde in the presence of ATP and NADPH. And we could use the wild type, which has activity for terminal alkenes, to assess molecules like that. And we could use our engineered catalyst to assess unactivated heteroaromatic like that, and we can functionalize heteroaromatics. Now, we designed a pathway to start, and we use one reaction as a model reaction to investigate our concept. And we plan that we can start with alkene or a terminal alkene like styrene, and use bicarbonate as a source of the CO2. And we can fix the CO2 to generate our carboxylate, express the CAR in whole cell, and we can use the CAR as whole cell preparation and assess our aldehyde. And because this is in whole cell, we have endogenous E. coli alcohol dehydrogenases, we can reduce this to the alcohol. And so we try this, we assemble the catalyst and the reagents that are needed, and we put everything in one pot. We change the concentration of the bicarbonate to see, because ideally we desire that the reaction to occur at near stoichiometric amount of bicarbonate. So ideally we want, we don't want to use a lot of bicarbonate. And interestingly, we observed that with just between 40 to 100 millimolar of bicarbonate, you could achieve almost full conversion. So starting from that, you could achieve that product in close to complete conversion. So this is a chromatogram. So this is a starting material with bicarbonate as the CO2 source. And we could generate an intermediate. Usually the acid is formed here and the aldehyde is formed here. And this is the product that we get. So starting from there, we get this product accumulated after 6 to 24 hours. We decided to use this principle to diversify our product profile. And we know that carboxylic acid reductase, if you, in the absence of NADPH, can catalyze the amidation of carboxylate to give you an amide. So we exploited this system, and we could use one buffer, so ammonium bicarbonate, as both the CO2 source and the ammonium source, and we could generate that to this. We could also branch this aldehyde to make amine. So once we go through this step, instead of going through the alcohol, we can incorporate an amine reductase or reductive amine acid in this system. And if you supply an amine source like the cyclopropylamine, you could generate the amine product through reductive amination. So a cascade of CO2 fixation, carboxylate reduction, and reductive amination to assess the high here. Of course, you could assess the alcohol. We also exploited our mutant to look at heteroaromatics like that. And so we can use our engineered mutant to assess the product's profile, of course, with the different skeletons, the same product and functional group, but with the different skeletons. I'd like to summarize that we have been able to exploit the power of cascade biocatalysis and semi-rational protein evolution to engineer enzymes and identify biocatalysts, and also assemble these biocatalyst lists to form an artificial pathway where we can now take simple starting material with CO2 and build more complex and value-added products. Now, the cascade reaction allows us to add value to the process. So you're starting from a very cheap material and you're getting something like that, or intermediate. But also, keenly and importantly, it allows us to address the equilibrium issues associated with fixing CO2. Because once you can withdraw the intermediate carboxylate to the next product, which of course is even higher in value than the carboxylate sometimes, you could achieve the full conversion of, you could functionalize any heteroaromatic aromatic systems or heteroaromatic to give you the corresponding carboxylate derivatives. And we exploited our detailed understanding of the kinetic of the reaction, as well as the structural and mechanistic insights from the reaction. With minimal screening, we're able to engineer the enzyme that would normally take acrylic acid derivatives. And we now have access to more interesting scaffolds like the heteroaromatic on unactivated aromatic systems. And using this engineer, we can expand our product portfolio, really. We can expand the product profile of our concept. We can start from terminal alkenes, but we can start from unactivated heteroaromatics or even potential aromatic systems. And we can functionalize this and assess different varieties. Depending on what enzymes you include in the next step, you can branch your product profile to a wide range of products. I'd like to end this talk here. And before I finish, I want to thank Professor David Lace in Manchester and the groups of David in Manchester, because this work was done just before I moved to Cambridge. And it was published in July this year. And I received support while I was in Manchester from ERCC, BBS, SCA, and Shell. And now I'm currently a Leverhulme Early Career Fellow, and I received support from Isaac Newton Trust as well. Florence Group in Biochemistry in Cambridge is hosting my research, and I'd just like to thank the Florian and the group as well, and the University of Cambridge for all the support. I'm happy to take any question if there is. Thank you very much, and thank you for your attention.
Thank you for that talk I'm going to moderate the Q&A session. So if anyone on Zoom has questions, they're welcome to ask them in person or to put them in the Zoom chat box. And if you are watching from YouTube, please feel free to put questions in the comments box there and I will ask them. So we have a question from Zoom. Why did you choose to use the fungal variant of the enzyme?
Right, so we've looked at the E. coli variant. So we look at the UBD in E. coli. And unfortunately for the scaffolds that we wanted, the enzyme was not active. And it was very difficult to get in vitro data or activity with the enzyme. This class of enzyme, we normally have problem with the cofactor binding once you isolate the enzyme. And so this was the problem with E. coli. So you need to reconstitute the enzyme once you have isolated it with in vitro cofactor regeneration system. And so we wanted to avoid that. The fungal one were more robust. They have the cofactor bound, so we didn't need extra step of reconstitution. And they were very active towards the kind of scaffold we wanted. So that was why we chose the fungal variants of the enzymes, if that's clear.
Thank you. Yes, that makes sense. Another question from Zoom is, would you mind elaborating on the, for example, pharmaceutical products that could be synthesized more easily using your method? Would you see these pathways being introduced in vivo using synthetic biology methods?
Yeah, so first of all, the first question, yes, we could make a lot of pharmaceutical, like if you know anything about antifungals, there's what we call allied antifungals derivatives. So if we take this scaffold, we could use that as the alkylating agent and couple it with an amine. So if we can generate that, and then if we have an amine, we can use it as a skeleton, and you can build antifungal agents. We call allyl-amine antifungal agents. And I think I started developing the profile where we can use from CO2 to build antifungal derivatives or skeletons that can lead to antifungal derivatives just before I moved to Cambridge. So yes, you can apply this platform to do that. And yeah, synthetic biology approach, of course, is an option for us to look at assembling these different enzymes in a host system to do that. The limitation is whether some of these substrates may be toxic to the host. And the other limitation is how much you need to optimize the fluxes of the system to achieve the conversion you want to. One advantage with the in vitro system, you can use it as lysate, and you can optimize the concentration of each of the enzymes to meet your method. And so this is the advantage of sometimes using the in vitro system. But of course, I agree that with the right amount of effort, you can put this system in a host system, is like is done in synthetic biology.
Thank you for those informative answers. Are there any other questions, either from YouTube or in Zoom? There's one more from Zoom. If toxicity and efficiency are issues, would it perhaps be an option to direct the enzymes to different compartments within the cell to sequester them?
So we've not investigated this in host cells or the whole pathway in host cell. I know we have used Chi as host cell, and what we observe is that depending on the amount of bicarbonate you have, you tend to have inhibition on the Chi system in host cell. But we've not investigated the whole pathway in the host system. So the issue of toxicity, I was just probably envisaging that it potentially could be. But yes, I totally agree that you could compartmentalize it into a system where you can probably withdraw the substrate. I don't know whether so you need to find a fine balance between the relay one intermediate to the next step. And if the compartmentalization does not affect the relay process, then of course that could be an option for you to do. But you need to take consideration of the stoichiometry of the enzymes, which is important, but also how one intermediate is related to the next step of the reaction.
Thank you. I've also had thanks from the person who asked the question. They say that makes sense. So if there aren't any more questions, I will then hand over to Christoph to introduce the next speaker. And thank you again for your talk.
Don't know what just happened there with that strange noise in the background, but thank you for that talk, Godwin. Really enjoyed that, especially as a budding biochemist. So we'll now hand over to Susanne, who will be talking about. Structural surprises